Good morning. Thank you for joining us for our speaker series, Toxicology and Societies, the Impacts of Chemicals in Our Lives. I'm Ruth Sofield, and I'm an environmental toxicology and chemistry professor at Western Washington University. And my co-host is Tracy Collier, a toxicologist who's retired from NOAA's Northwest Fisheries Science Center and continues his work as a consultant. Hi, Tracy.
Ruth, good to see you.
Good morning. We would like to begin by acknowledging that we at Western Washington University are on the ancestral homelands of the Coast Salish peoples who have lived in the Salish Sea Basin throughout the San Juan Islands and North Cascades watershed from time immemorial. Please join us in expressing our deepest respect and gratitude for our Indigenous neighbors, the Lummi Nation and Nooksack Tribe, for their enduring care and protection of our shared lands and waterways. And now I'd like to go over a few logistics. We will save questions for the end of the presentation, but you can use the Q&A feature on your screen at any time. You will be muted throughout the presentation, so we will use this feature to interact. The focus of our speaker series is to provide understandable examples of how events with the toxicological component have consequences to people and societies. Today, we are delighted to welcome Dr. Deborah McKenzie, a research associate professor in the University of New Mexico College of Pharmacy. She is a co-director of the Community Environmental Health Program and one of the principal investigators for the Native Environmental Health Equity Center at the University of New Mexico. Today, she will present on her and her collaborators' work on the Navajo birth cohort study and the Thinking Zinc clinical trial. Welcome, Dr. McKenzie.
Thank you. Well, I do appreciate the opportunity to share with you the work that we do here at UNM with some of our tribal partners, focusing on our work today on some of the research that's ongoing on the Navajo Nation in partnership with the community. Okay. So the Community Environmental Health Program is a program that was founded at UNM by Dr. Johnny Lewis more than 30 years ago, with the idea to facilitate integration of population studies with mechanistic toxicology studies in order to identify strategies to deal with environmental exposures to toxic metals to impacted communities. And I'm going to give some overview and some background on some of these studies that I've highlighted here on this page to illustrate some of the work that we are doing. A lot of this work started in the human health studies began in 2005 with what we call the Dene project. Dene is the term that Navajo use to describe themselves, so the Dene Network for Environmental Health. The focus is on uranium exposures and chronic illness. And so the community was concerned about the impacts of living in proximity to mines and mine waste on chronic disease. And so this study was initiated initially with 1,300 surveys, health surveys, and followed up by some biosampling. And this was one of the first research programs to examine community impacts in partnership with Navajo chapters that within the Eastern Agency of Navajo Nation. Following this study, we initiated the Navajo birth cohort study. The study was initiated in 2010 as a result of community concerns and a congressional mandate to address the health concerns, and focuses on the impact of legacy or chronic environmental exposures to abandoned uranium mine waste on child health. And we will talk about some of our preliminary findings and our ongoing studies to address these concerns. We also have a Center for Native Environmental Health Research Equity that has been funded since 2015. This is a partnership with 3 different Native American communities, the Navajo, Sioux, and Absaluke Crow. And we're looking at a comparative study looking at health effects from metals and environmental exposures in different communities to understand some of the similarities and distinctions to identify the greatest risk. And I'm not going to talk about that study much today, but that is also an ongoing study. We also have a UNM Metals Superfund Center that has recently received our second round of funding. And this is a multidisciplinary and transdisciplinary team science research project, again, in partnership with Navajo as well as local Pueblo communities, to examine environmental health risk from mine waste to communities, and also to design and develop interventions to reduce and reverse the impacts of these exposures. And in our first phase of the Metals Superfund Center, we had a pilot clinical trial, and I will talk with you about that, give you an overview of what the purpose of the trial was and some preliminary results. So Native American communities who live within the Western United States have an opportunity to experience substantial exposure to metals and metal mixtures. And the Western United States is home to more than 50% of the US indigenous population, and also overlays with a considerable number of abandoned hard rock mines. So there's 161,000 approximately abandoned hard rock mines in this region, of which 5,000 are abandoned uranium mines. So it's estimated that greater than 600,000 indigenous peoples live within 10 kilometers of abandoned mine waste. And as I will talk about on Navajo Nation, much of this mine waste and mine sites are unmarked, unfenced, and easily accessible to individuals as well as livestock. It's estimated also that 40% of the surface waters in these areas are contaminated with metals and other contaminants from mining activity. So, as many of you are likely aware, Native American communities who live in these areas are potentially impacted in numerous ways. And one is that they live close to the land, so they have heavy reliance on local resources. Infrastructure is minimal in many communities. This impacts overall health, access to care, supplies, and other phenomenon. And Native American communities have been understudied and underrepresented in many health studies in the United States and other countries as well for a variety of reasons, which are complex. And we can discuss if anyone has a desire to do that. So on Navajo Nation, the last mine closed in 1986, but there are 500 abandoned uranium mines that remain today. And as you can see from this image on the right, it's difficult to discern what's landscape and what's mine waste. There are also 1,100 unremediated waste sites that arose from mining activity. And as I mentioned earlier, many of these are unfenced, unmarked, and not well delineated for the community members. So since mining activity, there have been more than 4 generations of community members on Navajo Nation who've been raised in the shadow of abandoned uranium mines, or AUMs. And this is an image of the Redwater Pond Road community, one of our partner communities, where there was a remedial action to remove the mine waste from the community. And you can see here they're scraping, this is the homes, and they're removing the 18 to 24 inches of contaminated soil. But the families had grown up and lived next to this mine waste for a long time, and it still remains, and remedial actions are still needed in this area, and these community members are still living here. So for us to really understand the health impacts of living in these areas and being exposed to these chronic conditions really requires iterative processes that incorporate environmental, experimental, and population studies. And that's what we try to do here at UNM in the Community Environmental Health Program. So we have a number of programs, studies that are looking at exposure sources, routes of exposure, characterization of the waste, looking at water, soils, plants, animals, trying to understand the exposure pathways. And we also have researchers who are using cellular, molecular, and animal models to validate and understand and identify mechanisms of toxicity. And then we combine that with looking within and conducting population studies to understand better what biomarkers, what changes, and what health impacts we're seeing from these different types of exposure. So it's an iterative and complex process and requires, as I mentioned earlier, a large team and integrative approaches. So from the Diné study, which was conducted in 2005 and 2013, we addressed two different types of exposure. And one was active mining area exposures. So we, through the surveys, questions were asked about active mining exposure, or exposure that occurred during the active mining period, which included not only the miners, but the family members of the miners, because the miners would come home covered in dust and their family members would wash the clothes. Apparently there were stories of kids who would take their family members or dads' lunch boxes and finish what food they may have had there. They either worked in some of the milling or mill reclamation sites or lived in proximity to a mining camp. And what we found, again, with the combination of the activity and the health survey, was that mining area exposures increased the risk of kidney disease in individuals who had this active mining exposure. And then we also asked questions about legacy exposure. So legacy exposure would be, again, not direct active mining exposures, but exposures that result from herding livestock, playing in and around abandoned mining sites, whether they may have known it or not. And what we found from this study, or this portion of the study, was that these legacy exposures increased risk for chronic diseases such as hypertension, autoimmune disease, and kidney disease, as well as some others. And we will talk a little bit more about that as we move through the discussion. So the autoimmunity that we noticed with this particular study was that individuals who lived in proximity to abandoned mines had a higher incidence of autoimmune, both biomarkers and self-reported autoimmune disease. And so biomarkers for autoimmune disease that we looked at in this study were autoantibodies. And so the odds ratio of having a self-reported autoimmune disease was overlaid with proximity to the mine sites. And I'd like to point out also on this page, you see this beautiful art. We have within our program a accomplished Native American artist from Zuni Pueblo who translates our art into science for the community members. It's very helpful to have this beautiful imagery so that we can discuss some of these complex science in interpretable ways. And this is a companion piece to immunity, and this is autoimmunity, trying to describe how your immune system can accidentally or unintentionally attack itself as opposed to fighting off pathogens. So I will have a number of her pieces scattered throughout our talk, and we are quite pleased to have the opportunity to have her beautiful work as part of our program. So as I mentioned, we're not just looking at population studies, but we're looking in cellular models to try to better understand what effects we see from metals and metals exposures. And one of the studies that was done again from samples from this original Dinesh study was that serum from the participants were co-cultured with coronary endothelial cell line. And then we were able to look for changes in biomarkers of effect or inflammation. And so what we were able to see is that the individuals who lived, again, in close proximity to the mines, I'm just going to click this down, had increased levels of pro-inflammatory endothelial markers stimulated in these cell lines. So there were factors in their serum that interacted with these endothelial cells that produced a strong inflammatory marker. And chronic inflammation is known to play a role in atherosclerosis through stiffening of arteries. And we also saw in these studies that uranium exposures were linked to increased C-reactive protein, which is another inflammatory marker, suggesting again that proximity as a proxy for exposure, as well as uranium exposure measures themselves, are associated in this case with increased inflammatory responses, which underlie numerous types of chronic illnesses. So an additional study was conducted using mine dust. So this was dust actually collected from the mine site and instilled into an animal model. And again, we were able to replicate some of the potential mechanisms consistent with cardiovascular disease, in this case, increased contraction and decreased relaxation of vessels in response to this mine dust. And this response was also reiterated using purified uranium and vanadium. So vanadium is a metal that's found in mine waste, also frequently in a compound called carnotite with uranium. So the health studies that we conducted in this Eastern Agency again provided some evidence that these legacy exposures do have health impacts. And so the community expressed concern about, well, if it's affecting our adults, what might it be doing to the health of our children? And so in 2010, the Navajo birth cohort study was initiated to address the impacts of uranium exposure on child's health outcomes. And this is a fairly comprehensive study where we enroll women during pregnancy and collect data on occupational history, activities, family histories of exposures, perform a home environmental assessment. So someone would go out to each home, measure indoor dust, look at water sources, location of the home, conduct radon surveys, gamma surveys, and additional information from the type of home, heating, water sources, etc. And we also collected blood and urine to perform biomonitoring. This was conducted in partnership with the CDC, and they analyzed our samples for a 36 element panel of metals, metalloids. And we collected samples at both enrollment and delivery during pregnancy from 250 dads when mom is enrolled, and then collected cord blood at birth and samples at 2 to 6 months and at 12 months of age. And we are now following these children up into the age of 9. So this study in 2016, the Navajo birth cohort study, became part of the NIH Environmental Influences on Child Health Outcomes National Consortium. And what this study does is it combines birth cohorts from across the United States in order to collect the same information to understand more broadly the impacts of exposure on pediatric outcomes. And this includes upper and lower airway outcomes, obesity, pre, peri, and postnatal outcomes, neurodevelopment, as well as positive child health. So we are now part of the ECHO program. So we now call ourselves NBCS ECHO. And this broadens our data collection and extends our follow-up for our children. So we now, again, enroll women during pregnancy, conduct surveys and questionnaires and biomonitoring, both at enrollment and at delivery for both mom and baby. And now we're able to also assess child health from birth to through 9 years of age now. Many surveys and questionnaires. We have over 100 different assessment tools covering exposures and outcomes, including biologic, behavior, psychosocial, medical, societal, and physical and chemical. And neurodevelopmental assessments are conducted by our clinical team between the ages of 3 and 5. And again, that should be between the ages of 7 and 9. And we conduct annual ages stages questionnaire. So that's a developmental screener. So our NBCS study right now includes participants that represent a cross-section of Navajo population. So all of our participants come from across Navajo Nation in both mining and non-mining chapters. So you can see the distribution of mine sites are the red X's, and then our participants are scattered across Navajo. And again, for those who may not be aware of Navajo Nation, there are parts of Navajo Nation in New Mexico, Arizona, and Utah. We have a team of almost 20 Navajo women and men who are our research field staff who live and work on Navajo Nation, and they are our partners who recruit and interact with study participants within their own communities. And they work, again, in close partnerships with both the IHS hospitals as well as the PL38 hospitals on Navajo Nation. So what do we see? So this is an old slide, but we still continue to see that the uranium exposures or the concentrations of uranium in our participants exceed national norms. So our studies show that the median urinary uranium concentrations exceed national norms by a significant amount. So 36% of men and 26% of women have urine uranium levels that exceed the NHANES value, which is determined by CDC. And the overlay here, so the bar shows the national 50th percentile, and then this is our population overlaid NBCS females and NBCS males. Primarily, I'll be focusing on uranium and arsenic because these are two metals of concern for their toxicity within this community, but we do have data on, as I mentioned, on 36 different metals and metalloids, and we'll talk a little bit about zinc and iodine as well. So arsenic is kind of interesting in this population in that if you look at the distribution for total arsenic, we look pretty similar to the national norms. But what we do see is that the ratio of organic arsenic to inorganic forms are different in our population, and that we have a higher level of the organic or the inorganic forms, which are more toxic, so the arsenite, arsenate, versus the organic forms. And organic forms of arsenic are found in seafood and rice, and those are not food sources that are common on Navajo Nation. So we still do find that arsenic, even though on the face of it, total arsenic doesn't look that different, there are still some concerns regarding toxicity of arsenic in this population. So getting back to, what some of our questions about the impacts of these exposures on birth outcomes and child health, we have looked at this in a number of ways. And one of the most compelling findings that we have so far is that when you look at metal mixtures and individuals that are exposed to multiple metals, we find that, again, individuals that have higher exposure to mixtures have an increased risk of preterm birth. So if you look at this, this is a clustering technique that classifies individuals into quartiles of exposure, and then uses a Bayesian statistical method to cluster individuals into groups. So if you're in cluster, if you have a quartile 1, you have a lower level of exposure, and quartile 4, you'd be in the upper level of exposure. So clusters 5 and 6 represent individuals that have higher levels of exposure. And if you look at the table down here, you can see that the relative risk in comparison to the reference group, which is cluster 1, is increased for preterm birth, suggesting again that exposure to metal mixtures does have a potential detrimental effect for birth outcomes. And the percentage of our population that falls in these different clusters, this analysis is 417 mothers are listed here. So about 20% of the population at this point in this sampling time have relatively low exposures. So we also have data that shows that prenatal exposure to uranium can occur during pregnancy. So in a study of 52 mother-baby pairs, we were able to identify that 25% of babies had uranium concentrations that at birth were above the NHANES 50th percentile value for adults, and that 92% of the babies with detectable uranium at birth were born from mothers who had uranium concentrations that were again above the NHANES 50th percentile. And that as mothers' concentrations increased, babies' concentrations also increased, suggesting again that we are getting exposure to uranium that occurs to the child during the pregnancy. So developmental exposures or developmental outcomes are one of our key outcome measures and concerns. And so we were able to show in this publication that prenatal exposure to lead, arsenic, antimony, barium, and copper, as well as molybdenum, predicted deficits in at least one of the ages and stages questionnaire inventory domain scores. So these analyses were done based on maternal exposures and micronutrients, and assessing in early childhood. So this is infant development. So this is again within the first year of life. Ages and stages questionnaire as an early developmental screener. We also noted in this study that mothers with lower SES status had higher levels of lead, cesium, and thallium exposures compared to mothers with the higher SES backgrounds within our population. One of the outcomes that we've observed with our neurodevelopmental assessments is a fairly high incidence of verbal delays. And statistical analysis demonstrates or shows that disparities in SES, so income, unemployment, and younger age of the mom are major contributors to verbal delays in children 3 to 5 years of age. But one thing that we were able to note through multiple statistical approaches is that micronutrient deficiencies in both zinc and iodine are also associated with verbal delay. And this is a finding that is of interest because this is a potentially mitigatable an opportunity to introduce an intervention, dietary intervention that can relieve some of this potential verbal delay. So we know that iodine and zinc are deficient in pregnant women in our cohort, as well as in the population as a whole. We'll talk a little bit more about zinc in a bit. And so we also see, on top of the sociodemographic risk factors, as well as the micronutrient risk factors, that toxic metals such as strontium and uranium also are additive effects on verbal delay. So there's still concerns with combinations of contributors to child development. So I've talked a lot about the data that I've shown you so far has focused a lot on maternal biomonitoring and maternal metal levels and prenatal and birth outcomes and child development outcomes. But we're also concerned about ongoing exposure for the children. And one of the things that we have seen is that our longitudinal analysis, so we're collecting data from delivery through 9 years. I have data on this graph that shows results from 5 years. And that by age 3 and 4, children are reaching adult concentrations of both uranium and total arsenic. So this indicates potential lifetime exposures, and that has a lot of concerns regarding chronic health conditions and increased rates of cancer in this population. And so it's important to have the opportunity to validate and establish that these exposures are occurring early in life and that they continue. And so we can begin to really elucidate and understand what role this plays in chronic health conditions that are of concern in our communities. So again, the data here is showing the ME is mothers at enrollment, mothers at delivery, and dads at enrollment, and then comparing the babies at birth, 6 months, 1 year, 2 year, 3 year, 4 years, and 5 years. So we also show on this graph, the dotted line shows you the NHANES 50th percentile and the 95th percentile. And you can see that many of our children are above the adult values for the 95th percentile at these early ages. So one of our goals then is to identify interventions that reduce risk. And I see that I'm going to have to hurry and continue through this, and we can do that. I want to talk to you about one of our clinical trials, and that is what we call Thinking Zinc. This is a nutritional intervention to mitigate the impacts of metal toxicity. And again, you can see our beautiful artwork by Ms. Mallory Kitaki and our pictures of our team here. And so why zinc? Why do we choose zinc? There's a number of reasons. And one is that zinc binding proteins are regulators of cell function. There's over 3,000 or 30,000 actually, zinc binding proteins in our body. And both uranium and arsenic have the ability to displace zinc in zinc binding pockets in certain proteins and disrupt their function. But if you supplement zinc and can compete back and restore function. And so we hypothesized that zinc supplementation could mitigate some of the health impacts and toxicity enacted by having higher levels of arsenic and uranium and bring the system back into balance. So the goal of the Thinking Zinc was to determine whether dietary zinc supplements reduce human disease that's induced by metals, and to conduct a clinical trial. So our biomarkers include autoimmunity, immune cytokines, immune cells, as well as DNA damage and DNA repair protein activity, because one of the major targets of our zinc binding protein that we know is targeted by arsenic and uranium involve DNA repair proteins. So this study, we enroll individuals, take 2 baseline samples from each individual, supply zinc, daily zinc at the USDA recommended daily allowance of 10 milligrams per day. And they take that for 6 months. And we again do measurements at 2 measurements post zinc. So we have 2 pre and 2 post for each individual. And our initial studies were done in the Redwater Pond Road community, which I introduced to you a little earlier. And a couple of interesting findings so far. And one is that when we look at longitudinal assessments of exposure, what we find is that we have fluctuations in exposure. So we have individuals who can be low one day and high other days. And so this is very informative for a number of reasons. And one, it makes us rethink a little bit some of our potential modeling or our cell culture models or animal models, in that typically chronic exposures are modeled by low dose daily exposures and not by sporadic high dose exposures. And it could be toxicologically quite different to have higher dose exposures followed by low dose exposures. So we need to think about how to model that in an experimental model so we can understand the health impacts of sporadic high dose exposure as opposed to a steady chronic low dose exposure. And also it allows us the opportunity to think about, well, what activities or weather or conditions resulted in a spike. So we would like to include some diaries, pull in our GIS team to look at weather and location, so that we can understand, the more we can understand about what causes spikes, then we can potentially introduce mitigation or interventional strategies to bring those spikes down so that we can reduce exposure in our population. So these are interesting findings, and we are following up on these. And our last data that I'm going to show you is that one of the biomarkers of DNA damage and repair, which is 8-OHDG, is decreased post-zinc. This is a molecule that is upregulated when you have oxidative stress and DNA damage. And so we're seeing that in this zinc trial, that we see a decrease in oxidative stress markers associated with this pathway following zinc supplementation. And so that's some positive data that we've received from our clinical trial. This is an ongoing trial, so sample analysis is ongoing, and we are working on finding funds to expand and continue this study because we believe that a dietary supplement is a potential widespread potential for a effective therapeutic approach to mitigate some of the toxicity from metals. Finally, I just want to say that we have a very large team that contribute to this work. This is the NBCS ECHO acknowledgements. Our Navajo team members are in turquoise colors. The bold indicates current, and non-bold are former team members. But we have a huge group of people who are committed to this study. And this is the Superfund acknowledgements. The clinical trial is funded through the Superfund mechanism. And with that, I will stop sharing and entertain questions and discussion.
Thank you so much. So I have some questions, and I wanted to start with one from our co-host, and that is, should we all be taking 10 milligrams of zinc per day? And I just wanted to say that I, from personal experience, had to look into zinc and its effects on humans, and I believe the difference in dose between therapeutic and toxic is pretty small. And so I'm just wondering, like, as a widespread, in a general population, widespread practice, what would you recommend around taking zinc? And then what do you think about with this population?
Zinc, I don't, from the data that we've looked at, the 10 milligrams and the toxic dose are not very close. So if you look at most of the supplements, they're at 100, 200% of the RDA, and we're at exactly RDA.
Okay.
Zinc is highly beneficial for immune function. So I would suggest that zinc is beneficial for a number of us. So zinc deficiencies are not uncommon, and especially in seniors. And there's a lot of studies that show that zinc supplementation can increase resistance to infection as well as other activities. So I would suggest taking zinc. I take zinc.
Okay.
But don't with any of these vitamins, taking excessive amount of vitamins is never a good plan. So, try to find vitamins that are as close to RDA, or find foods. I mean, that's one of the things that we do with our community members is we talk about what kind of foods are zinc rich. And so as it turns out, blue corn mush, which is a native food that's consumed during pregnancy, especially if it's made in the traditional way, which includes some ash preparation, that is a good source of zinc. And so we talk about that. And in fact, we have one of our field staff members will bring blue corn mush to community meetings and pass it around and talk about different foods that are available and consistent with Navajo diet that are high in zinc. And so that's one of our educational activities is to discuss, okay, well, if zinc is going to help you, what is a natural way to increase your zinc?
Okay. And I just want to say, like, it's really neat to see the range of what you all are doing, from mechanistic understanding, monitoring, all the way to intervention. So it's really impressive, and I appreciate that you're presenting all that for us. And so in that, for you, what's the most rewarding aspect of all of this, if you think about the range of what you've been involved in?
Well, I grew up in a lab, my early years, of science, I was always doing studies with human cells, and, I always wanted to know what was going on in the body. But I really do find that working with the community and working with I guess my biggest enjoyment is working with our research field staff, because they live and work in these communities. They are so committed to our study participants, to the moms, to the babies, and just seeing that. And we have staff that have been with us since the beginning, and they're so vested in this. And it warms my heart to know that they're helping their community and they're part of this, and this study is theirs. So.
Cool. We did have a question from a community member about why there's been the focus on the Navajo Nation. And I wonder if it's more than just proximity. Is that the main reason, or is there more?
There's a little bit more. So the birth cohort, to do a population study, you have to have a relatively large population. And so Navajo Nation is one of the largest tribal communities. They have their own institutional review board for human studies. So there's a mechanism to interface with them. So all of this work is approved and overseen by Navajo Nation. And all of the data that I have presented to you and all the information I've presented to you has been presented to them and approved by them before ever presenting to outside. So that we and data sharing agreements are all maintained and protected under tribal sovereignty. So, those are reasons, part is that, those were where our partnerships began, but we do have projects that include, as I mentioned, Cheyenne River Sioux, Crow Nation, and a number of the Pueblo communities here in New Mexico. One of the Superfund focuses heavily on Laguna Pueblo. So the Laguna Pueblo is close to what was one of the world's largest open pit uranium mines. And so there's a different type of exposure there. And so we're not focused entirely on Navajo, but I focused on that for this because otherwise it gets a little bit hard to follow. So while the Navajo birth cohort study, again, because we had a large enough population of births to be able to conduct this study. So.
Okay. You mentioned earlier in your talk that Native American populations are understudied. And would you comment further on that and tell us?
Yeah, they're historically understudied in these larger health studies for a number of reasons. And it's not a blanket reason because each community is different. I think there was a question about epigenetics also in the comments and one of the things is that there's currently a moratorium on genetic research on Navajo Nation. So we're not allowed at this point to conduct any of the genetic analysis. So some communities are more open to that than others. And tribal sovereignty and data sharing issues are, again, need to be negotiated and established with each community. Some communities have a better structure for doing that than others. And so each time you work with a community to address their concerns, there are individual tribal community considerations regarding privacy, data sharing, data ownership. And sometimes researchers aren't familiar with these different considerations. And so it's establishing a relationship with each community, and that takes time and it takes effort. And it's I think improving, but it's something that has to be considered and be done again at to the community's satisfaction, that they know that their wishes and their data ownership are respected.
Yeah. Maybe on a slightly different note, so the uranium mines, who's responsible for cleaning those up, and then also for any impacts to humans and the environment? Who's responsible for that?
So many of these are unclaimed, unmarked, and nobody has responsibility per se. There are some that are under cleanup. So the EPA and the Navajo EPA work together to work on cleanup. So there are some sites that are have some remedial action, but it's slow and tedious work. So cleaning up the mess will not be something that's done in our lifetime. So that's why some of these mitigational strategies and understanding what is causing these spikes in exposure are going to be ways that we can reduce risk, that are going to supersede a cleanup effort.
Yeah. Are there examples in your time where you've seen improved health outcomes occur? And you showed a little bit with the zinc clinical trial that supports that there could be improvement through that type of intervention. I'm also thinking, like, if there were a site that were remediated and you could show some before and after improvements, is that something that you all are thinking about or able to measure? Or
We're thinking about it. We don't have data that supports it at this point because we don't have enough information on really what the exposures are coming from. So, you think, okay, well, it's going to be water, or it's going to be dust, or it's going to be this or that, but it's a combination of. And so we're trying to really determine what the best approximation of where do you start with that. So unfortunately, we don't have enough information to show a before and after for health effects, and the health effects are still so ill-defined. So really understanding how that happens is not well.
so, I mean, that's amazing because you've been working on this for how many years now?
We know it causes inflammation. We know it
Right.
does, but again, it's like if it contributes, I think the metals exposures clearly contribute to chronic disease, but the straight line is not straight.
Right. So do you need more funding, more time, more
Yes.
people, more? Yeah. Okay.
Yes, and it's frustrating for the community members because you go out and you're doing a study and they're like, you guys have been looking at this for a long time. I want answers, and it's like, well, we want answers too. It's hard to explain to a community member why science is so darn slow. And that's you asked me what my greatest pleasure or success or what I feel best about. That's what I feel worst about, is that it's hard to explain to them why we don't have answers.
And how do you do that? What do you say?
I don't have a good answer for that. I wish I had answers, and I don't. It's hard to go out there and say, we're trying to find answers, and they've been living with this for, a long time. They want answers, and they deserve answers. And it's, too little, too late sometimes. And so, we do the best we can, but I still feel like we don't and can't do enough. It's hard to do enough.
Yeah. We have we had a couple of questions related to, what can we do? And one was in the form of, are there legislative reps that we can contact, or broader public health policy changes? Like, so there's the science and what we can do, but what can happen from kind of more of a federal policy regulatory aspect?
Yeah, and those are good questions. And what we need is the data to establish that, yes, these exposures are occurring. Yes, they're associated with these health encounters. So that's step one. We work closely with Navajo government, with the Navajo Health and Human Services Council, with the Navajo president, vice president. We have a partnership with the Navajo Department of Health. And so a number of our research field staff are actually employed by the Navajo Department. So we have a sub-award with them. So we have these partnerships with Navajo Nation, and to inform them as soon as we find stuff. We also work with Navajo EPA, and with EPA again. And I think the funding agencies, NIH and NIEHS, have supported this research and are continuing to support research in tribal communities. And so I think that's, advocating for more funding at both a congressional level, as well as because they're the ones who determine the funds And again, at the political level is helpful.
Mhm. I have a question about a slide you showed, and it was the arsenic levels in males and females. And it was interesting that there was, what I saw looked like a difference in the distribution where the men had higher concentrations and kind of a narrower distribution. Is there an explanation for that? And
I don't really have an explanation for that other than we have fewer males than we have females in our study right now. And I'm not sure whether it represents, a real thing or whether it's because of metabolic processes. Again, our mothers are pregnant, and pregnancy we know affects some of the metals levels. And so that's a confounder in trying to make that direct comparison. So that's a potential question of metabolism, of exposures, activities coming in more in contact, eating more food that has, so there's numbers of reasons, and we don't really have an answer for that, but there are reasons why there might be differences.
okay, final question. If you just get the answer to anything related to the work you do, just get the answer, what's the one question you would like an answer to?
Are you asked?
Yeah, like if you as you think about, so I was just thinking, okay, why is there a difference in the male versus female arsenic? And so what's the one thing that you just say, I wish I knew this answer?
So one of my key interests is metabolic syndrome and diabetes. And I would like to know what impact early life metals exposure has on increased risk for diabetes, because diabetes is a huge problem in our communities. And there's more and more data that suggests that it's possible that a connection between especially arsenic exposure and diabetes. And so that's the answer I would like to have, because I think that would if we could mitigate that in some way, even through zinc, reduce that burden of diabetes on this population, that would be a huge benefit. So that's, as a biochemist and person, that's kind of where that's the answer that I think would have one of the biggest health benefits on a younger population that would affect adult health as well. So that'd be my question.
Excellent. All right. Well, I want to thank you for presenting. Really interesting and informative, and just appreciate you being here. So thank you so much.
Well, thank you for the invite, and I appreciate that. So.
Yep. All right. Cheers.